I want you to do something right now. Just take a look at your own hands, maybe flex your fingers a bit, make a fist, and then open it back up.
It's a pretty standard movement, right?
Exactly. But every single movement you just made and, every thought you were having as you process my words, it's all powered by these microscopic cellular engines deep inside your body.
Which we take completely for granted.
Oh, 100%. We rarely think about where they came from. But if we trace the evolutionary bloodline of those tiny power plants keeping you alive this exact second, we hit this bizarre and honestly startling paradox.
Yeah, it genuinely is one of the most profound twists in all of biology.
Right, because they share a direct ancient ancestor with one of history's most devastating, ruthless bacterial killers.
You have this foundational mechanism for complex life, and its closest known relative is a pathogen that literally wiped out armies.
Which is exactly what we are unpacking today. Welcome to the deep dive. We are looking at the fascinating, microscopic, and historically deadly world of a bacterial genus known as Rickettsia.
And to really wrap our heads around what Rickettsia actually is, we kind of have to look at why it confused scientists for decades. I mean, early 20th century microbiologists were convinced this thing was a virus.
Wait, really? But if it has a cell wall and its own DNA like the basic hallmarks of a bacteria, how could they possibly confuse it with a virus?
Well, because of how it behaved in the lab. Rickettsia is what we call an obligate intracellular parasite.
Meaning it absolutely cannot replicate outside a living host cell, right?
Exactly. If you take a standard bacterial pathogen like E. coli or strep, you can just swab it onto a Petri dish full of nutrient jelly, put it in an incubator, and watch a colony grow.
Right, the classic high school biology experiment.
Yeah, exactly. But Rickettsia will just die. It needs the living machinery of a host cell to survive.
Yeah.
So because it was incredibly small and because it failed to grow on those standard lab media, researchers at the turn of the century just lumped it in with viruses, which also require host cells.
So they were judging it purely by its behavior rather than its actual biology.
That's the core of it, yeah. We know now it is a true bacterium. It has its own ribosomes, its own DNA, its own RNA. It's technically classified as gram-negative, which is a structural category for bacteria, but it doesn't even play by those rules properly.
Let me pause you there on gram-negative, because that usually means a bacteria takes up a specific dye so we can actually see it under a microscope, right?
Normally, yes. A standard gram stain turns these bacteria a pinkish red. But Rickettsia stains incredibly poorly. It is almost invisible under normal protocols.
Oh, wow. So how do you even find it?
Microbiologists actually have to use specialized chemical stains or tag the bacteria with glowing fluorescent antibodies just to prove it's even there in a tissue sample.
Okay, so if you're an early 1900 scientist, you can't grow it on a dish and you can barely even see it under a microscope. How on earth did they figure out what they were dealing with?
They had to use animal models and, well, tragically themselves.
Right, because they couldn't isolate it safely on a glass dish.
Exactly. They had to handle highly infectious blood and tissue. Which brings us to the really tragic origin of the name itself. The genus Rickettsia honors an American pathologist named Howard Taylor Ricketts.
I know he traveled out to the Bitterroot Valley of Montana between, I think, 1906 and 1909.
Yeah, he set up this makeshift laboratory out there in a tent, of all places, to investigate this terrifying, frequently fatal illness the locals called black measles.
Which we know today as Rocky Mountain spotted fever.
Right. And Ricketts was working with incredibly primitive tools by our standards. He didn't know the exact taxonomy of the pathogen, but he managed to prove that you could transmit the disease to guinea pigs and monkeys via infected blood.
And didn't he also find the bug that was carrying it?
He did. He made a massive breakthrough when he identified wood ticks as the vector carrying the disease.
He actually figured out a mechanism called transovarial transmission, right?
Which was totally revolutionary.
Ugh.
Normally, with a vector-borne disease, an insect has to bite an infected animal, pick up the pathogen, and then bite a human.
Like a mosquito with malaria.
Exactly. But Ricketts discovered that an infected female tick passes the Rickettsia bacteria directly into her own eggs.
So the offspring are born already carrying the biological weapon.
Yes, it's a literal inherited family curse for the ticks.
That is wild. It completely broke the known rules of infectious disease transmission at the time.
It really did. It meant the ticks weren't just a delivery vehicle. They were a permanent living reservoir for the pathogen.
So his success in Montana must have made him the absolute go-to expert for this kind of mystery.
It did, which unfortunately led to his final assignment. In 1910, Ricketts traveled to Mexico City to study a massive outbreak of a completely different disease, which was epidemic typhus.
Okay, a totally different disease, but same bacteria family.
Exactly. He successfully identified the causative organism in the blood of patients and traced it to body lice. And then, tragically, he contracted typhus himself.
Oh, man.
Yeah. He died in Mexico City on May 3, 1910. He was just 39 years old.
He dies, not even knowing that he had uncovered an entirely new class of bacteria. And he wasn't the only one who paid that price, was he?
No, the occupational hazard of studying this organism claimed several lives. In 1916, a Brazilian physician named Henrique de Rocha Lima formally proposed the genus name Rickettsia.
To honor Ricketts.
Right. De Rocha Lima was in Europe studying typhus during World War I. He wanted to honor Ricketts, but he also needed to honor his own research partner, a Czech scientist named Stanislaus von Prohacek.
Let me guess, another casualty of the lab?
Sadly, yes. Prohacek died of typhus in 1915 while they were investigating an outbreak in a German prisoner of war camp.
Wow.
So de Rocha Lima named the specific organism that causes epidemic typhus Rickettsia prohaceki.
I want you to think about that for a second. The entire taxonomic foundation of this field is a monument to martyrdom. Both the genus and the species name for epidemic typhus memorialize two scientists killed by the very organisms they were trying to understand.
It is a really sobering piece of medical history.
So it is incredibly deadly to researchers in a lab setting. But when a tick actually bites someone in the wild, or a louse infects a soldier, what is the bacteria physically doing once it gets inside the body?
It targets a very specific tissue, which is the vascular endothelium.
And those are the delicate single-cell linings of your body's capillaries and small blood vessels, right?
Yeah, exactly. Unlike pathogens like tetanus or cholera, which just sit in your body and act like little poison factories pumping out toxins, Rickettsia physically invades your cells.
It breaches the cell wall and basically sets up shop inside.
It does. And the mechanism it uses to spread is both brilliant and bizarre. We see this specifically in the spotted fever group, like the bacteria that causes Rocky Mountain spotted fever.
Okay, so what does it do?
Well, inside a host cell, the cytoplasm is full of these floating protein building blocks called actin. Rickettsia grabs those host proteins and polymerizes them, essentially snapping them together like Legos.
Like it's building a structure.
Yeah, it builds a rigid propulsive comet tail behind itself.
Wait, so it's stealing the structural scaffolding of our own cells, building a ladder behind itself, and using that to physically push itself forward.
Yes, it generates enough physical force to shove itself against the host cell's membrane, push right through it, and punch directly into the neighboring cell.
It is a microscopic bank robber. I mean, imagine a crew robbing a bank vault. Instead of running out into the street to get to the bank next door, where the cops, or in this case, the immune system, can see them, they just tunnel through the basement wall straight into the adjacent vault.
That is a perfect analogy. Because they do that, the bacteria never actually exposes itself to the bloodstream. They stay entirely off the immune system's radar.
That is terrifyingly smart.
And the clinical consequence of all that tunneling is widespread destruction of the blood vessel linings. This causes systemic vasculitis. Your capillaries literally become compromised and leaky.
Which explains the signature body-wide rash we see in these diseases. The rash is essentially blood leaking out of damaged microscopic vessels under the skin.
It's basically a whole body microhemorrhage. And because blood vessels are everywhere, this vascular damage quickly leads to multi-organ failure. The kidneys, the lungs, the brain, they all start shutting down as their blood supply gets compromised.
And how fast does that happen?
Fast. Before antibiotics, untreated Rocky Mountain spotted fever had an estimated case fatality rate of 20 to 25%, often killing the patient within a week or two.
So the mechanism of tunneling through blood vessels is consistent across the genus, but you mentioned different diseases. How does one bug cause local sporadic outbreaks and another wipe out entire populations?
It all comes down to the insect delivering the payload. Taxonomically, the genus is basically split into two clinically essential branches. First, you have the spotted fever group, which is transmitted by ticks.
And those are the localized ones. You literally have to walk into tick habitat to get it.
Right. Then you have the typhus group, which includes Rickettsia prohaceki. These are transmitted by body lice and fleas.
And a body louse behaves very differently than a tick waiting on a blade of grass.
Radically differently. The human body louse, Pediculus humanus corporis, lives in the seams of clothing. It thrives in cold, crowded, unhygienic conditions where people just can't wash or change their clothes.
So refugee camps, trench warfare, prisons.
Exactly. When people are freezing, they huddle together for warmth, allowing the lice to crawl easily from person to person.
And the louse itself is reacting to the disease too, right? Because the sources mention that when a human host gets a high typhus fever or, their body goes cold after they die, the louse senses the temperature change and flees to find a new, healthy host.
Right, the insect's basic survival instinct accelerates the epidemic. This dynamic turned typhus into a literal, historical decider of wars.
Let's talk about that, because the historical impact is staggering. Let's look at Napoleon's invasion of Russia in 1812. He marches in with his massive Grande Armée, roughly 600,000 men.
And we always hear about the Russian winter defeating them. But the winter just forced them to huddle together in dirty uniforms. It was typhus, along with dysentery, that actually decimated the ranks. Historians estimate disease killed far more of Napoleon's soldiers than actual combat with the Russian army did.
A microscopic bacterium turned a military campaign into an epidemiological catastrophe.
And it happened again on a massive scale a century later. During World War I and its immediate aftermath, the League of Nations estimated typhus caused 3 million deaths in Russia and Eastern Europe.
3 million. That is just difficult to even process.
It is. The sheer scale of it prompted physician Hans Zinser to write his famous 1935 book, Rats, Lice, and History. His thesis was that epidemic typhus shaped the outcomes of wars and the borders of empires just as much as any general or politician ever did.
But the tide did eventually turn against the disease, specifically during World War II, thanks to a massive logistical intervention in Italy.
Yes. In late 1943, an epidemic was raging in Naples. Allied forces realized that if typhus got into the troops, it could literally jeopardize the European theater. So they deployed a brand new chemical compound, which was DDT powder.
The logistics of this are wild to me. How do you delouse an entire city?
You set up dusting stations all over the place. Soldiers literally took manual pump dusters, like the kind you would use in a garden, and pumped clouds of DDT powder down the collars, up the sleeves, and into the waistbands of civilians' clothing.
Wow.
They dusted over 1 million people. It was one of the first massive, successful uses of a chemical agent to break a human disease vector. The epidemic collapsed almost overnight.
So you would think that after dusting entire cities and fighting this pathogen for over a century, if I walk into a clinic today with a tick bite, modern medicine would have this perfectly figured out.
You would certainly hope so, but diagnosing rickettsial diseases today is actually still full of perilous clinical traps. The biggest issue is what doctors call the spotless fever diagnostic trap.
How does that happen?
It traces back to how we actually test for it. Historically, doctors relied on something called the Weil-Felix test, which was developed in 1916. It exploited a really bizarre biological coincidence. Rickettsia bacteria share a similar carbohydrate molecule on their surface with a completely different, harmless gut bacteria called Proteus.
Wait, why does the body confuse two different bugs?
It is basically a lock-and-key mechanism. Your antibodies are the keys designed to fit specific locks on a pathogen surface. Because Proteus and Rickettsia have similar locks, antibodies built to fight typhus will accidentally bind to Proteus.
Okay.
So doctors would drop Proteus into a patient's blood sample. If the blood clumped up, it meant typhus antibodies were present.
That is a clever workaround for the time. But we have modern immunofluorescence assays now. Are those foolproof?
Surprisingly, not during the first critical days. Both the old Weil-Felix test and our modern tests look for the body's immune response, the antibodies, not the bacteria itself.
Oh, I see. It's like a security system. The test is looking for the security guards, the antibodies, not the intruder.
Exactly.
In the first week of infection, the body hasn't deployed the guards yet. So the security cameras show an empty room, and the test comes back negative, even though the intruder is actively breaking into the vault.
That is a perfect analogy. The tests frequently come back completely negative during the first week of illness. And to complicate things even further, an estimated 10 to 15% of Rocky Mountain spotted fever patients never develop a rash at all.
Or the rash starts hidden, right?
Yeah, it starts on the wrists and ankles and gets misdiagnosed as just a generic viral illness or an allergic reaction.
So if the tests are negative and there is no rash, but a patient comes in with a high fever and a terrible headache after hiking, what is a doctor actually supposed to do?
The CDC protocol is urgent and definitive. Treat first, confirm later. You immediately administer a cheap, highly effective antibiotic called doxycycline. You do not wait for the rash, and you do not wait for lab confirmation.
Because delaying treatment by even a few days is the single strongest predictor of death.
Exactly.
Which is so counterintuitive, because we are so conditioned to think doctors need to know exactly what is wrong before prescribing antibiotics. But here, excess caution can literally be fatal.
And historically, excess caution did cause fatalities, particularly with pediatric patients. For decades, doxycycline and other tetracycline antibiotics carried really strict warnings against being used in children under the age of 8.
Right, because older data showed prolonged use of these drugs could cause permanent tooth discoloration and enamel issues.
Exactly. So physicians were afraid to prescribe it to kids. They would withhold the drug or use less effective alternatives when a child came in with suspected Rocky Mountain spotted fever. And you have to keep in mind, this disease disproportionately hits boys aged 5 to 9 in endemic areas.
The tragedy there is just infuriating. Holding back the one drug that reliably cures a fatal disease over cosmetic fears of stained teeth.
It is heartbreaking. It took years of follow-up studies to prove that the short courses of doxycycline used for rickettsial diseases, which is usually just 5 to 10 days, do not actually cause significant tooth staining.
So the guidelines finally caught up.
Yeah, around 2015, the CDC and the American Academy of Pediatrics finally revised their guidance to explicitly recommend doxycycline as the first-line therapy for children of any age.
So the treatment is sorted provided you catch it early. But we need to address a massive geographic misnomer. The name Rocky Mountain spotted fever gives people a very false sense of security. I mean, I live in North Carolina. I hear Rocky Mountain, and I think I'm perfectly safe.
But today, the vast majority of U.S. cases aren't anywhere near the Rockies. The disease is heavily concentrated in the south, central, and southeastern United States. So North Carolina, Oklahoma, Arkansas, Tennessee.
And the sources also highlight a completely new, ecologically distinct outbreak happening right now in Arizona.
Which changes the entire risk profile of the disease. In Arizona and parts of northern Mexico, the vector driving the outbreak isn't the classic wood tick or dog tick you'd pick up hiking through a damp forest. It is the brown dog tick, Rickettsia sanguineus.
Why does a different species of tick matter so much to human risk?
Because the brown dog tick can actually survive indoors. Most ticks need the high humidity of leaf litter to survive. They dry out and die inside a house.
But not this one.
No, the brown dog tick has adapted to infest homes, dog beds, and dry yards directly. You don't have to be out trekking through wilderness vegetation to get infected. You could just be living in a suburban house with dogs and poor yard maintenance.
So we have this stealth tunneling bacteria that evades our tests, changes its vectors, and is now literally moving into our living rooms. Why is it so uniquely elusive? I mean, why is its biology so strange compared to a normal bacteria like E. coli?
Because over millions of years of evolution, Rickettsia has engaged in extreme biological outsourcing. It has essentially thrown away its own biological toolkits to rely entirely on the host cell.
It is the squatter in a furnished house. Someone breaks in, moves in, and immediately throws away their own oven, their own refrigerator, their own bed. Because why lug all that heavy stuff around when you can just use the host's appliances?
That is exactly what happened at the genetic level. Because it lives exclusively inside the nutrient-rich soup of host cells, Rickettsia doesn't need to synthesize its own energy. It literally steals ATP, the energy currency of the cell, as well as amino acids and nucleotides directly from the host.
And if you don't need the genes to make those things, you lose them.
We see the evidence of this genome shrinkage really clearly. An average free-living bacterium, like E. coli, has a genome of about 4.6 million base pairs. In 1998, a team led by Siv Anderson at Uppsala University finally sequenced the genome of Rickettsia prowazekii.
And what did they find?
They found his genome was tiny, only about 1.1 million base pairs. It had discarded the vast majority of its genetic baggage.
But the real bombshell wasn't the size of the genome, it was what that genome actually matched.
Yes. When Anderson's team compared that shrunken, highly specialized sequence against all known DNA, Rickettsia prowazekii emerged as the closest known bacterial relative to mitochondrial DNA.
Okay, let's ground this for a second. The mitochondria are the powerhouses of the cell. They are the organelles generating the ATP that allows my heart to beat and my brain to function. You are saying that the biological machinery keeping humans alive shares a common ancestor with the bacteria that causes deadly epidemic typhus.
It provided the massive molecular proof needed for Lynn Margulis's endosymbiotic theory.
Walk us through how that theory works. How does a pathogen become a powerhouse?
So, billions of years ago, before complex life existed, a primitive eukaryotic cell, or an archaea, swallowed a Rickettsia-like bacterium. Normally, the larger cell would just digest the bacteria for food.
Right.
But in this one miraculous instance, the bacteria survived inside the host. Perhaps the host cell was starving and the bacteria was leaking extra ATP. They essentially struck an evolutionary deal. The host provided a safe, nutrient-rich home, and the bacteria provided limitless energy.
And over millions of years, that bacterium lost its independence entirely and became the mitochondrion. Meaning that complex cellular life, the very basis of human existence, the reason we can breathe oxygen and grow past the size of a single microbe, essentially began with an ancient parasitic infection.
The microscopic origins of all complex life are inextricably tied to the same biological family as one of humanity's deadliest historical plagues.
It fundamentally changes how you look at the world and your own body. But there was one final fascinating thread hidden in the taxonomy that I want to explore before we wrap up because it points to the future.
Yeah, Rickettsia belongs to a broader order called Rickettsialis. The taxonomy has seen a lot of recent shakeups, splitting off organisms that cause scrub typhus and Q fever into different branches. But there is another distant cousin still residing in the family tree called Wolbachia.
And Wolbachia doesn't infect humans at all, right?
No, not at all.
Yeah,
It infects the reproductive tissues of a huge percentage of the world's insects. It aggressively manipulates their reproduction to ensure its own survival. And recently, scientists discovered something incredible.
What's that?
When a mosquito is infected with Wolbachia, the bacteria outcompetes human viruses inside the insect's body.
Wait.
Yeah. Today, scientists are deliberately infecting mosquitoes with Wolbachia in the lab and releasing them into the wild. Those mosquitoes breed, passing the bacteria to their offspring. And a mosquito carrying Wolbachia is physically incapable of transmitting deadly human viruses like dengue fever or Zika.
The biological poetry of that is astounding. The very same evolutionary family tree that gave us epidemic typhus, the plague that decimated Napoleon's army and ravaged trenches in World War I, is now being actively weaponized by humanity to eradicate our worst mosquito-borne plagues.
Yeah.
We are literally using one parasite to block another.
It perfectly captures how deeply entangled we are with this invisible world.
That is an amazing thought to leave you with. Next time you flex your hand, take a breath, or, swat away a mosquito on a summer evening, just remember the invisible, complex, and sometimes deadly evolutionary history that makes all of it possible. Thanks for joining us on this deep dive. Keep questioning the invisible world around you, and we will see you next time.